One of the things that we notice with humans is that our traits um, that particularly are prevalent in anatomically modern humans are not selected for only by kind of ecological circumstances, but also through the role that culture plays. So, for example, if we're just looking at strictly uh, anatomical traits, right, the fact that we are seeing an increasing proportion of the population that doesn't have wisdom teeth is kind of allowed for by the fact that we cook our food and we no longer have selection favoring that third set of molars erupting around the age of 18 or 19. There are other traits that going to correspond to some of these cultural differences. I mean, as a whole, right? We lost hair pretty early. We were able to lose hair in this context of tropical development, but once we started moving outside of the tropics, we needed to compensate through the use of clothing and such, and that's going to play a factor even in this selection for lighter skin outside of the tropics. So we can't discount the role that culture plays in shaping our biological evolution. So we have to approach anything that addresses biological variation from this biocultural perspective. Um, so we're going to look at two specific examples, that of the sickle cell trait, or maintaining the allele for sickled red blood cells because of the innate resistance to malaria that it gives us. This is not something that emerged until relatively recently, within about the last 2,000 years. So evolutionarily, we would have spent very little time in areas that had a lot of mosquitoes. And we even look at modern tropical foragers, and their response to mosquito bites is very, very different from how like American grad students going down to tropical areas, how they respond to mosquito bites. So one of the things that has shaped this dynamic between us and malaria is the fact that we develop food production. In particular, it's the shift towards what we call swidden agriculture. That is the small-scale horticulture that we see in dense tropical rainforest, where we've got slash and burn of fields. We've got pools of standing water. We don't use a lot of irrigation with swidden agriculture. So looking at basically creating this breeding ground for mosquitoes that are then going to expose us to malaria. DNA studies have identified a recent origin and spread of the sickle cell trait in a specific population from Senegal and West Africa, with the trait appearing sometime between about 2100 and 1250 years ago. So our innate immunity to malaria had experienced strong selective pressures to be able to become as prevalent as it is in these areas with a lot of malaria carrying mosquitoes. And this is something that's useful for us to consider because we are seeing a rise of mosquito-borne illnesses. And so now we talk about things like I can't pronounce this one. Chikungunya. We talk about things like the Zika virus. We are going to, with climate change and more areas becoming kind of subtropical or tropical, we're going to see an increase in malaria-borne illnesses. The selection pressure then for this sickle cell shape to red blood cells is the advantage that it gives us in this innate kind of immunity to malaria. Lactose tolerance, or what we call lactase persistence, is another trait that has emerged fairly recently. And we point to Middle Eastern herders that moved into Europe about 5,000 or 6,000 years ago as kind of the drivers for the development of lactase persistence. When we look at modern herding populations, there's a coevolutionary response between herders and their livestock. And so herders select for livestock that have higher quality milk production. That consumption of milk then selects for herding populations that have increased lactase persistence. This is not something that only occurred as herders moved from the Middle East into Europe, but we see the kind of independent and much more recent evolution of lactase persistence in Africa. Interestingly, herding is something that we find across much of the Eurasian steppes into areas like Inner Mongolia. It's not always associated with lactase persistence. So we see some areas of the world that have incredibly high lactose intolerance, including Asian populations. So there's a fundamental difference in populations that consume their dairy products in a fermented form than those that consume milk itself. And so, in areas like Mongolia, a lot of the dairy consumption is through the form of cheese and yogurt, which that fermentation process helps to break down the lactase that's present in those milk products. So we can see a difference based on kind of microecology, right? And based on something cultural like how we prepare our dairy products or process our dairy products. So here we just have some images, one that represents the life cycle of the malaria parasite, showing that it has kind of two stages. It's got its stage that's within the mosquito, and then the stage that's within the human bloodstream. And then here on the right, we've got a picture of a Fulani woman, not Fu- no, a Maasai woman who is milking cattle, demonstrating um, the consumption of these raw dairy products. And then we have a young child with the sheep or the herds of goats in the Gobi Desert. In areas like the Gobi Desert, we don't have lactase persistence because those milk products are consumed in a fermented state. One of the ways that we identify the kind of path that evolution is taking is through population genetics. Population genetics is an area of research that examines the allele frequencies in populations and attempts to identify the factors that cause allele frequencies to change over time. And so if you remember early on in the semester, one of our definitions of evolution itself was simply change in allele frequencies or gene frequencies over time. This is where population genetics really makes important contributions. When we look at populations, we sometimes define them as the group within which individuals find mating partners. We can call that group the mating pool for particular individuals. This is sometimes determined by things like geographical barriers, where we may develop breeding isolates. Island nations, for example, are common breeding isolates because it's hard to get between them. It's you've got a smaller population in a finite geographical space, and so their breeding pool becomes restricted almost by necessity. But there can also be social things that circumscript breeding populations. So, religious taboos, for example, the caste system in India, where we've got really strong social pressure to only marry and reproduce within your caste. So, what these allow, these breeding isolates, is the opportunity to study microevolution, small scale evolutionary change between isolated populations. Once a specific human population has been identified, population geneticists will examine their allele frequencies, compare the observed frequencies with those that would be predicted by equations like the Hardy-Weinberg equilibrium equation. So we make predictions based on the relative frequencies of the homozygous dominant, the homozygous recessive, and the heterozygous recessive individuals. And so we expect things like this equation p squared, that's homozygous dominant, plus 2pq, which is heterozygous, plus q squared, that's homozygous recessive, equals 1. We expect for allele frequencies to follow this general rule of thumb. So when we find incredible differences, then we have reason to suspect natural selection favoring of the particular expression of the particular genotype that produces a phenotype. So much of human variation is adaptive. To survive, organisms have to maintain normal function within this context of ever-changing environments. Much of what we do, both behaviorally and physiologically, is a response to stressors. One of the big kind of underlying factors that we're not really aware of because it doesn't involve conscious choice are the physiological processes that maintain what we call homeostasis. Or this kind of functional response, this constancy, this internal constancy. So adaptation as a functional response is in response to these environmental conditions that individuals and populations experience. Our adaptations may be genetic, particularly those that are solutions to very old adaptive selection pressures. Bipedality, for example, is genetic, it's not developmental. But they may also be more short-term and indicative of what we call acclimatization. Acclimatization is a physiological response to changes in the environment that occurs during an individual's lifetime. We can also acclimate to conditions on several different temporal scales. So, for example, we can have ontogenetic adaptation, where the way that the conditions that we experience during growth and development change how our body grows and functions, and those are irreversible changes, so that your anatomy, your physiology is always going to be indicative of this environment in which you developed. We can also have very short-term physiological responses, and we'll talk about some of those with respect to our adaptations for dwelling in high altitude, kind of hypoxic, low oxygen environments. So we will start with our discussion of solar radiation and skin color, because when we talk about race, that is one of the, if not the most visible phenotypic feature that we, particularly in the United States, classify racial categories on. Prior to European contact, we observed what was called a cline, which means just predictable geographic variation in skin color based on latitude. So populations had the greatest amount of pigmentation in the tropics. Lighter skin then emerged as we moved away from the tropics, both into more northern latitudes and to southern latitudes. Our skin color is produced by melanin. This is a granular substance that's produced by melanocytes in the epidermis. We all have the same number of melanocytes, so we don't see a difference between populations in the number of melanocytes they have. What we see instead are differences in the amount of melanin that the melanocytes produce and the size of the melanin granules, so that people with darker skin both have melanocytes that produce more melanin and have melanocytes that produce larger melanin granules. Melanin is adaptively significant. It has an important role acting as a built-in sunscreen, because the melanin granules absorb UV radiation. And what this does is protects us against mutations in the skin cells that could turn into things like skin cancer, melanoma. Exposure to sunlight triggers melanocytes to produce melanin. This is a protective response. So the more sunlight exposure that you have, the more your skin cells are going to produce melanin, which means the darker your skin is going to get. We call this kind of short-term response tanning, for example. And we recognize that different populations have different responses of their skin cells to high levels of solar radiation. So we look to some of the genetic changes that have given us things like red hair, fair complexion, and freckles, right? People who are redheads tend to burn rather than tan. So this is a function of their melanocytes and the amount and size of the melanin that they produce. So in an evolutionary context, darker skin would have been favored. Early hominins lived in the tropics, right? There are many animals in the tropics that have lighter skin but darker coats. But even among those animals, chimpanzees included, areas of their bodies that don't have hair, that are exposed to higher levels of solar radiation, those areas are lighter. So if we look at chimpanzee faces relative to um, chimpanzee torsos, for example, the skin on their faces is lighter than the skin on their torsos. I mean, darker than the skin on their torsos. And this helps to protect them against things like sunburn. Um, so the fact that we lived in an area of the world where solar radiation would have been a sizable factor demonstrates that, at least initially, um, selection for dark skin outweighed other kinds of functions of lighter skin. When we emerged as a species, we were all dark skinned. Early hominins were also constantly exposed to solar radiation. This is different than how we live in a modern context. Many of us don't spend the peak hours of the day, let's say between 10 and 4. Many of us are indoors during that time. I mean, think about the jobs that you have. If you're working in an office, for example, you're not out in the sun with this constant UV exposure. Then if you're like a weekend warrior and spend a lot of time outside on the weekends, you end up with sunburn when you increase that UV exposure. Early hominins also didn't wear clothing that would protect them from solar radiation. So as we lost our body hair, which is a function of adapting to a really hot environment and being able to effectively cool ourselves, as we lost that body hair, we increased the selection favoring dark skin because we didn't have any protective benefit of our hair against the solar radiation. So our bodies were experiencing greater exposure. UV radiation, thusly, is a powerful, or was a powerful, selective agent, selecting for high levels of melanin production. There have been some proposals that by like 2050, the average skin color is going to be more like a hot chocolate mixed with milk versus the degree of super light and super dark skin that we see around the world. And part of what's driving this change is um, more acceptable genetic admixture between racially separated populations. But part of what is driving this change is also the kind of loss of this selection pressure favoring darker skin. So, I mean, we spend our days inside. When we are outside, we are either wearing clothing or we put on sunscreen. Both of these reduce the selective pressure that UV radiation exerts upon our melanin and our melanocytes. Melanin is also important in preventing the degradation of folate. Folate is critical for women of reproductive age because it prevents neural tube defects. This can include things like spina bifida, or cases where part of the spinal column or even part of the brain occur outside of the neural tube or the spine. In areas with high UV radiation, we find that lighter skinned individuals have much lower circulating levels of folate because that folate is destroyed by the solar radiation. So an additional selective pressure favoring darker skin in tropical areas, areas with high UV exposure. Lighter skin at higher latitudes was selected for as populations moved outside of Europe. It is a response to these new selection pressures that include things like cold temperatures and cloudy skies. These cloudy skies are not just seasonal, they occur both in the summer and winter. That means less solar radiation is actually reaching the Earth. These colder temperatures are also then associated with increased clothing wear because you needed to cope with cold. We also have then shorter day length during certain seasons of the year, particularly during the winter. So cumulatively, this leads to a lot lower exposure to UV radiation. So instead of melanin providing a protective benefit against high levels of UV radiation. In far northern latitudes, lighter skin was selected for so that individuals could produce enough vitamin D, right? We need vitamin D. We are actually at a point in modern times where many people are vitamin D deficient because we've got the message that once we go outside, we need to have sunscreen on, and that message has been received so effectively that people are interfering with their body's natural ability to produce vitamin D. We're not really able to absorb vitamin D very effectively from our foods. The vitamin D that we produce through sun exposure is a lot more, let's say, bioavailable, right? It's better for us. It's easier to get vitamin D from the sun than it is for us to absorb it through food. So insufficient vitamin D results in growth abnormalities and malformation, including things like rickets. This is where legs are often bowed because there's not enough vitamin D for healthy bone development. We did find in relatively recent historical times that African Americans had a higher incidence of things like rickets because their vitamin D production was lower. This was ameliorated during like the 1950s and 60s through fortifying dairy milk with supplemental vitamin D. So it factors into some of the public health decisions that we've seen even in our country. So it wasn't so much a factor that that dark skin wasn't favored in higher latitudes, but more of a function of, I mean, it's bidirectional, right? That if you had dark skin in higher latitudes, you were more likely to demonstrate these developmental abnormalities like rickets. But if you had lighter skin in tropical areas, you were more likely to sunburn and then have associated mutations that lead to things like skin cancer. So we've got simultaneous selection for dark skin in tropical areas and for lighter skin in farther northern areas. Now, one thing to note is that some relatively recent headlines, both with respect to Scandinavians, there's a documentary out on the first Swede, for example, and to populations in the British Isles, that light skin color is a relatively recent phenomenon. There is a fossil that's nicknamed Cheddarman in Great Britain that was fully sequenced genomically now and has alleles that code for dark skin, not light skin. Similarly, this documentary on the first Swedes, though I haven't seen it in its entirety yet, talks about how early on in Swedish kind of inhabitants that the early Swedes were also very dark skinned. So our kind of Eurocentric idea of light skin being a European trait is a relatively new phenomenon. And this vitamin D deficiency becomes even more prevalent with the advent of agriculture because we plant products don't produce a lot of vitamin D. You get more of that from animal products. And so when our diets shifted more to more heavily dependent upon agricultural goods, we also experience lower intake of vitamin D rich meats.